Hi everybody, this is Honors Bio 2 Mechanisms of Cancer, and this is the second genetics or inheritance talk that I'm giving. I had a lot of trouble with this talk because I think that inheritance is generally oversimplified and kind of taught a little incorrectly. I think the old way of thinking about things is that you have for example, a flower or plant, and it has two versions of a gene, a purple one and a clear one, and you call the purple one big P and you call the clear one little p, and then if you have those two alleles of that gene together, then you have a purple flower, and if you have a little p and a little p, it's a clear flower. And that is certainly how the way it works for that gene, but there's a lot of problems with that. First of all, I think with new technology, we don't just look at flowers anymore. Flower color is not something that we just examine. We can see actual receptors on the surfaces of cells now. So the idea of phenotype being what something looks like has changed dramatically, because it's not just what something looks like with your eyes anymore. It's something that you can see under a microscope. So last time we talked about how new genes were made, parts of a gene, and why are we doing that? Okay. How to make new genes. We talked about how genes were transferred from me to you. If I'm making a clone, it's through cell cycle and mitosis. And if I'm trying to make a new baby so that it's got genetic differences from me, we're doing it through cell cycle and mitosis. And that brings us to basics of genetics. Okay, so the idea behind all of genetics and basically all of biology is that it's, by solving these problems, we come to the future. We generate new therapies, new cures, and everybody's happy. And we keep thinking that, which is kind of funny. And that was certainly the idea when these two guys went head-to-head. They fought over the Human Genome Project. The guy on the left, his name is Craig Venter. The guy on the right is Francis Collins. They're still both around, so this isn't too long ago. I think the Genome Project was finished in 2000, which would make it ancient for you. But these two guys were in a race to solve the human genome. And again, they thought if they solved the human genome, then you would get new therapies and new cures and everybody would be happy. But it was kind of an interesting race because Craig Venter is head of the Venter Institute, and they do really weird things. So, for example, Venter has made a genome, and he put it into a bacteria, and so he's essentially, well, they've claimed to make artificial life by that bacteria actually replicates now, is alive, and it makes proteins, and his daughter's name is in the protein. So is his dog's, I think. I think he has two daughters and a dog or something like that. And their names are spelled out in the bacteria's proteins. And kind of that's a good way to market them or tag them so that you know that they're his bacteria. So any bacteria with that protein, with the daughter's name protein in it, would be his. Had he won the Human Genome Project or sequenced the Human Genome Project first, I think some interesting things would have happened, because everything that this guy published or found, he had to publish because he was head of a public effort. So it kind of stunk, but everything Francis Collins had, Venter could get a hold of. But Francis Collins had also the joint work of many scientists from around the world and the funding of the NIH. So he could really get a lot done. You can read more about that down here in that link when you get this. But the Human Genome Project yielded the human genome. So let's go ahead and see what that looks like. This is the UCSC genome browser. It's basically Google for the human genome. So we can go ahead and search basically any genome we want. We can look for a bush baby, a naked mole, a pika, a megabat. Oh, that sounds awesome. A rock hyrax, a tenrec, manatee. You can search all these genomes here for genes that you want. And so let's go ahead and search for insulin receptor. Okay, so we have insulin receptor. First of all, the kind of disappointing thing that you find out right away is there's tons of different versions of everything that you want, so you can't figure out which one you want to look at. So we're just going to look at a random one. We're going to look, so these are all things, sequences of DNA that matched up to the insulin receptor in some part. So this information tells us a few things. It tells us that we're looking at chromosome 19. We're looking at the P arm, so this is a chromosome, and you can see this the centromere. The left side is called the P side, and the right side is called the Q side. I don't know why. And then that little red thing is 181,000 base pairs wide. So our gene is somewhere in there, which is 181,000 base pairs is actually pretty specific. So we're going to look at reference sequences, and then we are going to look at see, this should do it. Nope. Here we go. They keep changing the interface on me, so okay, so here's the whole gene, and the red is okay, hold on. So the G is a splice site. The red is transcribed. No, it's part of the promoter that is not transcribed. The blue is transcribed but not translated. The black is, no, I did that wrong. Okay. G is the splice site, A is transcribed but not translated, blue is transcribed and translated. So we're looking at the insulin receptor, and basically the exons are going to be blue and red. The introns are going to be everything else. So this is what the human genome looks like, and this is what the Human Genome Project yielded us. And as you can see from this, I don't know if you can tell, but if you look right here, you can tell that this is actually a beautiful brown-eyed baby girl. And if you look down here, you can tell that the baby girl is gonna like asparagus, and it looks like hang gliding as well. At the age of 36, they're going to do something important. I'm not sure what it is, but it's very important. I'm obviously being sarcastic here. Oh, there's a blue part. The point is, from this sequence, you can't get much. You don't know what any of this stuff does. This might all be part of another gene. This might be transcribed and then used in RNA interference. This could be for histone binding, but the Human Genome Sequence project only gave us letters. And you see, this is all intron. I just wanted to show you guys that. I think it gives you a better idea of a gene. And like I said, this is all available to you and it's public. So, okay, so we're back. I don't even know if you knew we took a break, but we did. So when I was trying to figure out how to teach you this, I went ahead and started looking through the newspapers, or actually online, and just found out what's being talked about as far as genetics is concerned. Okay, so this is a molecular knockout about Alzheimer's, and it's got an HDAC in it, which means that it's an epigenetic story. So a knockout is when you actually do fake recombinations. So you basically put a little piece of DNA into a cell with a fake recombination site, and it lines up. If you can do this, it takes a little trying, but you can get it to line up to, for example, the gene for the protein HDAC6, and then you put in a trigger for recombination, and the thing you put in will flip into the human genome, and the HDAC6 gene will flip out. Then you make that thing have a baby with another thing that you did the same thing to, so now the baby of those two has no HDAC genes, and we call that a knockout. It's basically a way of telling what the function of a protein is. So you make a knockout, and then you find out what's not working, and then that's the function of the protein. You should probably add that to your techniques part of your binder. Okay, so this is a genetic test for DNA based for signs of cancer. Notice they didn't say for cancer. Obviously, you can't generate a genetic test for cancer. Here's a really interesting one. It's the idea of putting genes into athletes to make really powerful athletes. Instead of giving them drugs, you actually give them the DNA and for whatever, growth hormones, or the ability to break down energy very quickly, the ability to clear out lactic acid very quickly. And is that legal? Can you make Olympians? Here's one that was about, I don't know why it's same-sex marriage, but it's about whether you can patent human genes or not. You can look that up. This one's back to epigenetics and Lamarck. I'm not going to go into that because you should be sick of that by now. This is a really cool idea. So it turns out that in voles there is a gene that you can use to predict monogamy. So basically, with certain levels of this protein, you can a vole will be monogamous, and without certain levels, it will be Randy. There we go, Randy. And for Michael Rothpletz, that's active. How do they term it? Active sex life. So that's kind of an interesting idea when you think about what genetics can influence. So I think we like to think that if we were going to cheat on a spouse or a significant other, that would be kind of a decision that we would make, and that it wouldn't be based on anything that was within us. However, this shows something different. The power of genetics on human thought is there's far more of an influence of genetics on human thought than I guarantee most of us give credit for at this time. Okay, so this is stem cells, and we've done that. And then this is 23andMe. 23andMe is where you can go to get your genome sequenced. And so I thought we would look at what your genetics can tell you. How or what can you know from a gene? Obviously, from the purple flower, we know that if we have the purple flower gene, then we get the purple flower transcript and the purple protein, and then we get the purple flower. This is a lowercase p, just so you know. So let's look at things that influence the process from gene to protein. No, from gene to effect. Okay, so we're going to try to go through a bunch of things that are affected in between genes and the effect of a gene. And like I said, you actually already know almost all of this stuff, so this should be pretty much a review for you. So let's go. Okay, so like I said, we're going to go, we looked at 23andMe just a second ago, and this is the kit 23 will send you, and what you do is you spit in it, and then you send it back to 23andMe, and then for $299 they send you this kit, and it says, hey, this is your likelihood of getting a disease. And if we went with the very, very simple model of genetics and the kind of the purple flower model, then we would just look for a dominant gene, and if you had the dominant gene, then you would have a disease. But in actuality, the information you get is it's not, hey, you have this gene, you're going to get this disease. It's a range. You get a range of percentages for most things. Like I said, you have about 20,000 genes right now, or 20,000 genes that code for proteins right now. And based on which ones you have, you can find your risk factors for a certain type of disease. But that's not whether or not you're going to get the disease. So why is that? Well, let's just remember all the different things that go into making a DNA into the effect of the DNA. So not just the protein, but what that protein actually does. So, oh, that's awkward. What we have here is a gene. You have the start of transcription here, you have your promoter here, you have your upstream activator here, and don't forget, you don't just have one upstream activator, you can have a bunch of them. So before we even get to transcribing the gene, we have to talk about transcription factors that are going to bind to the DNA, that are going to recruit RNA polymerase. Then we have to talk about these upstream elements, which can either which are going to either positively influence transcription or negatively influence transcription. And then, of course, we have to think about epigenetics. So this is an epigenetic map. Red means that something is on. Green means that it's off. I don't remember why they did it like that. I kind of think they should do it the other way. But you can see this is all one genome, and these are different patterns of expression. So different parts of the genome are turned on and off in all of these different cells. So before we're even talking about really the parts of the gene, the transcription factors, and the upstream activating elements, whether you can transcribe a gene is going to be definitely dependent on whether it's on, whether it's available, or whether it's epigenetically shut down. Okay, so then you have to look at, assuming that we transcribe something, assuming that it gets translated, what kinds of things can happen to proteins and what kind of relationships can they have with each other. So let's say we have a regular protein. Okay, so if it gets a mutation in it and it generates an allele, you could get one of those alleles. So you could have a wild type protein, and then you could have another protein that was different only by one mutation but caused a loss of function. Or you could have a mutation in it that maybe caused an early stop, which again caused loss of function. You could have mutations that cause gain of function, so this could do something completely different or do whatever it's supposed to do better. And then you could have conditional loss or gain of function things. So, for example, this protein will only function above 37 degrees Celsius. This one will function above 25 degrees Celsius. So if I have the 25 degrees Celsius one and the loss of function mutation together as my two alleles, it's a little bit more difficult to figure out, what the end effect is going to be. And just so you know, when you transfer alleles, you don't just transfer one allele at a time, you transfer allele blocks. So this would be one gene, and there's probably some genes here too. So crossing over is more likely to happen here, and we're going to transfer one of those blocks. I don't remember why I put the slide in. Okay, so we're going to blow through this, but this is going to be really ridiculously easy. So these are just going to be really basic interactions proteins can have with each other. So if I have two alleles and both of them work, then my whatever is going to work. Makes sense. If I have two alleles and one of them works and one of them doesn't work, well, I still have the one that works, so whatever is my system is probably going to work. And just because I have one allele doesn't mean that I'm going to have half of the proteins produced. It could be more, it could be less. You don't know. If I have two alleles, neither of them work, then you're kind of messed up. Here's another interesting relationship. Let's say we have a protein. We want to make one of these. So you need this part and this part. And they're both identical. If you have the two alleles, or two genes, two different versions of it, and they each make one side, then you're fine. However, if you have two versions and one makes a straight line and this one makes the regular one, then you could end up with one of these which doesn't work, one of these which doesn't work, and only when you have two proteins coming from this allele would you have something that worked. And you can figure out this relationship, by the way, with a plus b squared, and I'll explain all that later, but that's that Punnett square crap that everybody else is learning. We'll talk about that. Things get more complicated when you have 4 proteins making up, or 4 pieces making up a functional protein. Here you can see one small piece can inactivate the whole thing. So even though I have 3 functional pieces here, I have one piece that doesn't work. Suddenly my, oh well, yep. Suddenly my, the effect of this piece becomes much larger. And then of course you have many different types of interactions. For example, what would happen if you have a protein that only functions at 37 degrees coming from one allele and one that functions at 25 degrees? And then, of course, you have the idea that proteins don't just act alone, right? So it's not the purple protein in the purple flower. Here you have something called leptin, which is produced by fat, insulin, which is released in response to how much sugar you eat, ghrelin, which is response to how full your stomach is, and I have no clue what PYY is. However, all of these things affect this part of the brain, which affects how hungry you are. So if I mess with this protein, we might have a big effect, we might have no effect at all. It depends on how this protein, leptin, reacts with insulin, ghrelin, and PYY. So here, the relationship between gene, for example, the gene for leptin, the protein leptin, and the effect it causes is really just not simple. So, in conclusion, this idea of purple gene, purple RNA transcript, I guess the transcript wouldn't actually be purple, and then the purple protein is not usually that clear. This one is actually an example of a human case that is kind of like the purple protein. This is the Huntington gene, this is the Huntington transcript, and this is the Huntington protein. If you have this version of the Huntington protein, basically a piece that has 20 repeats of a inner segment, you're going to get the disease. But Huntington's is really rare in that it is one of those diseases, if you have the gene, you have the disease. So the relationship between gene and disease is very clear. We'll talk about this more next time. I've got a friend behind me.